This video is the take-home messages from the cell cycle, mitosis, and meiosis. Okay, so over here we have a life cycle, and that shows us how to go from fertilized egg to the adult organism. We need to do a lot of mitosis. And this is when we start with the cell that is a zygote here, and it is 2N or diploid, and then we go through many cell divisions, each time producing an identical diploid cell, and eventually producing an adult organism. Within that adult organism, in specific tissues, then gametes can be produced, and those are haploid cells, which are N. And if we have two different gametes that meet up, then they can go through the process of fertilization and produce a new diploid organism. So again, this is a life cycle. This is not the cell cycle. The cell cycle instead is this symbol here, mitosis, G1, S, and G2. And I'll talk about that more on a different slide. So that's the cell cycle, and this is a life cycle. Okay, so let's talk about the cell cycle, mitosis, and meiosis. So again, I start to draw these little symbols. So the cell cycle starts with G1, which is a gap phase or growth phase, followed by the S phase, which this is when DNA is replicated. Then we have the G2 growth phase, and then finally we have mitosis, which we start with prophase here, we end with telophase and cytokinesis over here. So basically the cell cycle, the big thing to see here is the cell cycle is more than mitosis. So mitosis is one part of the whole cell cycle. We also can call this whole thing here, this is interphase, G1, S, and G2. And this is when the cell doesn't really look like it's doing anything, but it's actually doing a lot in terms of getting ready, and as far as the number of copies of the DNA, that is very significant in the S phase. That's when we double and make an exact copy of each of the chromosomes present. Another thing to notice here is that basically what we're doing, this is kind of like the circle of life, if you will, for a cell. So a new cell is born up here at the beginning of G1. This would be a diploid cell. And basically then that cell is going to get larger through this process. It's going to duplicate its DNA through this part of the cell cycle. And then in G2 it's going to continue to grow larger. And then basically what mitosis is all about is splitting up those two copies of the genetic material. So mitosis, the whole purpose of that is to properly divide the copies of genetic material into two cells. And basically then that means that at the end of mitosis is really focusing on prophase, prometaphase, metaphase, anaphase, and telophase. So that's all about the genetic material. And cytokinesis is happening at the same time as telophase, usually. If it happens, there are some cells it doesn't happen in, though we're not going to talk about those much. But cytokinesis is actually cleaving the cell into two different cells. And so at the end of that, then we have two diploid cells. Two diploid cells. And these two cells are identical to each other. If one of these cells would then go through the process again of cell division, then that would go back into the cell cycle, and it would have to go again through all of these processes to get ready, and then eventually to divide the information into two cells. So a really important thing for this section is make sure that you know all the details about the cell cycle. So this whole thing over here is really important as far as when the DNA is getting replicated and when it's getting divided. And we do get a lot of press time to look at the different parts of the phases of mitosis, which we can see over here. Your book breaks it down into prophase, prometaphase, metaphase, anaphase, and telophase. And so the big things to keep track of here are the chromosomes, which are shown in blue. Then there are microtubules or spindle, which is shown in green. And it doesn't show these very well, but I'll draw a few of them in there. Let's see, what color should we do? I guess I'll just do black. And then we keep track of the centrioles as well. So the centrioles are at the top of each of the holes. That's where the microtubules will originate from. And there's like two little hollow parts right there. Those are centrioles. The centrioles would be up here, 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 here. So why we care about the centrioles and the microtubules so much is that this is how the chromosomes can move. So the chromosomes cannot move on their own. Microtubules are the part of the cytoskeleton which can actually take the chromosomes, kind of like a fishing line, and then move it around. Like if the chromosome were a worm, then the fishing line would be the microtubules. I should also mention that the cell cycle that I've drawn up here is not really drawn to scale, so mitosis is actually the shortest part of the cell cycle. Okay, let's talk some more about the names of the different chromosomes. So the terminology for this is can be a little hard to master, but it's absolutely crucial that you learn the terminology so that you know what you're talking about. Okay, so there's three major things to keep track of. Sister chromatids, these are identical copies. These are identical copies of a chromosome. If this was the original copy of the DNA, then this sister chromatid, the replicated copy, would be produced during the S phase of mitosis. And again, the big thing here is that these are identical. On the other hand, when we talk about homologous chromosomes, these are similar but not identical to each other. Your book will usually show these as being the same length and shape. They also have the same genes. For example, we may have the A gene here, and then we'd see the identical copy over here, but then we might see little a and little a over here. This would correspond to this particular region of the chromosome. They have the same genes, but across the homologous chromosomes, the dark blue versus the light blue, they may have different alleles. Now for the non-homologous chromosomes, we're comparing this long blue chromosome to this orange shorter chromosome. And so what they're trying to show you with that is that these are completely different chromosomes. They are different lengths and shapes, and they have different genes altogether. So the orange chromosome may have the D gene here, big D. This one maybe would have little d on it. And so they're just different letters of the alphabet. They encode totally different things. Okay, so now we need to check out how these different chromosomes will align as we go through mitosis and meiosis. These have major implications for whether a cell is haploid or diploid, and also has implications for if it has the identical information as all of the other cells produced through that process. Here we're looking at a single organism that has 3 different cells in metaphase. Each of these are the different phases of metaphase. For example, metaphase of mitosis here, metaphase of meiosis 1 here, and metaphase of meiosis 2 here. And so basically notice that we have a organism that has 2 different chromosomes, one long and one short. And in the metaphase of mitosis, we see that there are 2 different copies of this. And so there's a red copy that's long with its sister chromatid. There's a blue copy that is long, but it could be a little bit longer. Let's just make it so. And then we see that there's a short chromosome. Here we see the red one with its sister chromatid. And then we see there's a second copy of this, the blue copy of the short chromosome with its sister chromatid. And so we know this is diploid because we have the 2 different copies for each of the chromosomes. And basically, remember that as we look at the long versus the short chromosome, then those are non-homologous to each other, versus the long to the long here, those are homologous, and the short to the short are homologous to each other. Okay, so remember that with the cell cycle, that we are going to watch the cell as it divides up the material. It can either divide it in mitosis, or it can go through G1S, G2, and then go through metaphase 1, followed by metaphase 2. And so basically we're just taking some select snapshots here to look at the chromosome alignment. Remember that the M phase is all about dividing up the nuclear material. And remember that through mitosis that we start with a diploid cell and we end up with a diploid cell. Versus for meiosis, we start with a diploid cell and then we end up with a haploid cell. So we have less material as we get towards the end. Okay, so let's talk a little bit about the metaphase of mitosis. Basically here we're just making a duplicate copy of the cell and its genetic information. So every single one of the chromosomes with its sister chromatid lines up in a nice, what I call single file alignment along the metaphase plate. And then each of the sister chromatids gets pulled apart from each other and goes on to its own new life at this pole. And eventually we will have two cells, one that forms with this material and one that forms with the other copy. And these will be identical to each other. Okay, now let's talk a little bit about double file. So this is metaphase 1 of meiosis. And for meiosis, remember that we start off with diploid, but we end up with haploid. And so that means that we need to get either one copy, just one copy of each of the homologous chromosomes. And so in that case, the homologous chromosomes actually all line up together in what I call double file. And then at this point, actually a little bit beforehand if you read your book carefully, but when they're in this double file arrangement, then crossover can occur. So this is the only time that crossover can occur. And so this is basically the alignment of the chromosomes, which happens in meiosis 1, which allows crossover to occur when the chromosomes can be cut and then swap information. So you get partially red and some blue on one side, and then you get partially blue and then some red on the other one. Basically, as metaphase 1 proceeds, that means that of these two homologous chromosome sets, they're going to get divided in half here. The sister chromatids are still going to stay attached to each other, and then they will stay attached to each other until they get all the way through metaphase 2, which we see over here. And so let's say that this was one of the cells. It got the long red chromosome, and then it got the short blue chromosome. And so now, as we proceed from metaphase 2 to the end of meiosis 2, then the sister chromatids will be separated. And there would be another cell that would have the long blue chromosome and the short red chromosome. And basically then we get 4 cells at the end of meiosis, and so those would all have different combinations of genetic material, and they would either have the red long chromosome or the blue long chromosome, and likewise for the short chromosome, either red or blue. And so this is how we end up with haploid cells, and it has to do with this very special arrangement here. This is super crucial. I call it double file, though your book never calls it that. It's also random for the cell as far as which of these homologous chromosome sets will get moved to the left or the right, and this is actually independent assortment that's going on here, as the two different copies of the genetic information can rearrange themselves, and then with respect to the other chromosomes, then they can be packaged independently from each other. Okay, let's look at, this is a slide from 23 annotated lecture notes. This is looking at what I call team mitosis versus team meiosis. And basically, we start off with a diploid cell in mitosis, and then we make 2 diploid identical cells at the end of mitosis. For meiosis, we start off with a diploid cell, and then eventually we make 4 haploid cells, which are each different from each other. So we've got our pros and cons here, so to talk you through those. So for mitosis, we're making a perfect copy of the cell, this parent cell, and then we make 2 daughter cells that are identical, and we have 2 copies of each chromosome. So this is great for having a really nice system to get the exact same copy of the cells, but there's no variety. On the other hand, for meiosis, we can make 4 different cells from each other, those gametes, and then we can get this variation through several different possibilities, one of which is crossover, another of which is independent assortment, which I talked you through on the last slide. It has to do with the respective copies of the maternal and paternal chromosomes for each homologous chromosome. And so that's another aspect of variation. And then the final one is that this produces gametes, which only have half of the genetic information from each parent. This means that we could mix information from two different parents and thus get a different combination. So all three of these things will contribute to variety. And so this is really important for evolution, that we have variation across a population and to allow change to happen. So some of the cons of meiosis is mistakes can happen, for example, due to non-disjunction if an extra or missing chromosome is packaged into a gamete, and also haploid cells are not a functional organism. They need to merge together, especially for animal cells, to make an actual adult organism. And then together, both of these are cell division types that are required for the life cycle in animals, and then some other types of eukaryotes.